viewers, welcome to this particular lecture on microbiology. In this particular lecture, I shall be discussing the different aspects or pathogenesis of the dreadful bacteria of Streptococcus pneumoniae. Here I shall be discussing the discovery, the gradual development, systematic position, transformation, cellular characteristics, and also the different diseases it causes and also their control. To start with, how this particular bacteria was discovered. This bacteria was discovered jointly in fact, in United States of America by George Steinberg in the year 1881. He discovered it, and at the same time Louis Pasteur also discovered in France. At that point of time, the exact cell structure was not denoted. Later on, it was in 1920 that it was observed that it is not a single round bacteria. It is actually two rounded cells or cocci joined to each other. So this particular slide shows that how it looks like. It looks like two cells joined together, so it was called Diplococcus pneumoniae. Subsequently, it was in 1974, it was found out that this is not actually two cells. It is a chain of cocci, so it was accordingly called Streptococcus, and the name was changed to Streptococcus pneumoniae. Now, coming to a very important phenomenon which was denoted by Griffith. What Griffith showed in the year 1928, that this bacteria, which is commonly called pneumococci, was injected to rat. And accordingly, he discovered two strains, type 2R and type 3S. So it was observed when 2R was injected to rat, the rat was surviving. That means the rat was killing the bacteria or destroying the bacteria, and the bacteria cannot kill the rat. At the same time, when 3S pneumococci was injected, the rat died because of pneumonia. So naturally, it was the virulent strain and 2R was the avirulent strain. Then it was actually heat killed. You know, that particular 3S, which is the virulent strain, was heated at 120 degrees centigrade, 121 to be specific. And when it was injected, it was observed that the rat survived. There was no signs of pneumonia. But now what he did, he injected living 2R and dead or heat-killed 3S strain. The rat should have been alive, but the rat died. So he concluded, due to some phenomenon, this particular 2R has been transformed to 3S. This phenomenon was called transformation by Griffith, but he couldn't explain the exact mechanism. Later on, three different scientists, namely Avery, MacLeod and McCarty, in the year 1944, actually denoted the transformation principle. And they showed it is actually the DNA of the dead cell which is causing the transformation in the living cell, making it a deadly strain. And this was the experiment they did. You can see here there was a mixture of DNA and RNA from the 3S bacteria. It was treated with RNAs, so the RNA was not there. And then the DNA was added to the 2R bacteria and the plate grew. Here the 3S transformants were produced. Why? That means the RNA was not there. Because of the DNA, this 2R was surviving as 3S. But in the next case, when the DNase was used, DNase destroyed the DNA, so there was only RNA, and thereby the bacteria didn't grow as 3S. So thereby they proved it scientifically that it is that particular DNA of the dead or heat-killed 3S strain which was not being destroyed is actually transforming the 2R bacteria to 3S. So this was the mechanism of transformation. Now, this particular bacteria, as you can see in the fluorescent dye, they will exhibit fluorescence, and it is also having some capsule outside. But what is their systematic position? The domain is bacteria, the phylum is Firmicute, class is Bacilli, order is Lactobacillus. So they belong to the same order as Lactobacillus. And the family is Streptococci, genus is Streptococcus, and the species here I am discussing, Streptococcus pneumoniae. Now coming to their distribution. They are distributed along the entire epithelial tissue of the human body. It can be in the nasopharynx. You can see these are the bacteria stained red in the nasopharynx. Also, it can be present in the respiratory tract as a normal flora. It can be present in blood, and it can also be present in different other tissue like the liver, like the lung, like the general stomach, or even digestive tract, even in the buccal cavity. And they are capable of showing some distinct property. One of the important property is that they will produce pneumolysine, which is causing the lysis of hemoglobin. And they also produce an extracellular matrix, which is a polysaccharide commonly called adenosine. So thereby these two cells, even if they are single cells, will adhere to each other, producing a colony. This is the structure of the adenosine protein or glycoprotein, and they can induce septicemic condition in HIV patient. This particular HIV infected person, you can see this is the streptococcal infection and how the entire edema and redness of the leg has been there, and if this condition persists, ultimately the leg has to be amputated. So it's a dreadful condition. And you can see this is the typical lancet shaped pneumonia cells or pneumonia bacteria, and this is also under powerful microscope. This is of course light microscope. You can see that they are chain of Streptococcus. The bacteria can actually grow on blood agar medium, and the hemoglobin is degraded, and this particular hemolysis action is shown here in this plate, where the red hemoglobin has been discolored because of the destruction by the streptococcal bacteria. Apart from that, the bacteria has got some specific identifying action. First of all, they are typically round or lancet shaped. They are diplococci, that is in double, in couple, or they can be in chain, streptococci. They are gram positive. They show specific symptoms. They show alpha hemolysis, and most importantly, they are sensitive to optocin, which is a particular chemical called ethyl hydrocuprin, and this particular sensitiveness is only found in case of Streptococcus pneumoniae, but not in other species of Streptococcus.
Sir, I have a question.
Yeah.
How optocin induce sensitivity in Streptococcus pneumoniae?
Yeah. Optocin actually binds with choline in their cell wall and thereby destroys the cell wall, and that is why it is sensitive. It cannot grow in presence of optocin, so we call it optocin sensitive. Okay. Now coming to the next important aspect, which is the outer capsule. This is also a very important character with which we can identify the bacteria. The bacteria will have a definite polysaccharide capsule. You can see here, this thick capsule will protect it during unfavorable condition or even sometimes against the enzymes of the host. This is the genome of this particular Streptococcus pneumoniae. The genome shows around 2.1 million base pair, having maybe around more than 1550 genes, of which 154 genes constitute the virulome alone, and there may be 176 genes which are non-invasive, and about 10% variability is there within this particular bacteria itself, which make the strain variable. As I was telling you that this particular bacteria exhibits two important phenomena. One is it can produce a spore, another one is it can produce transformation. So here you can see in this slide, the sporulation is actually production of a specific endospore through the formation of pre-spore and forespore, and ultimately this is the structure which help them to survive during unfavorable condition, and the spore gene, SPO-A in fact, is responsible for induction of sporulation. But at the same time, when I was telling competence, this bacteria is exhibiting competence with COM-E gene. And with the competence, what is the competence? The competence will ensure that this bacteria will take up foreign DNA and will modify themselves. So this is how it takes up the foreign DNA and gets transformed. So transformation, it has been proved, actually requires or involves about 23 genes. And there are some specific molecules which can induce transformation. And this includes the mitomycin C, or it includes norfloxacin.
Sir, I have a question.
Yeah.
Is there any negative effect of induction of competence?
Yeah. When you are inducing competence, then it can become transformed. So it can become pathogenic. It can become deadly. So by competence, first of all, the bacteria can survive in oxygenic stress and at the same time it can become deadly. And the combination even becomes worse when this particular bacteria is resistant to norfloxacin. So unnecessarily you are exposing the bacteria to norfloxacin, it is resistant to norfloxacin, and at the same time it will show competence because of norfloxacin, so it becomes more deadly. So this is the negative aspect of competence in Streptococcus, right? Now there is another very important factor which I should have said, that is the activation of RECA. The RECA which is activated, the RECA gene activation during competence actually help them to survive in oxidative stress and oxidative burst. And thereby when the oxidative stress is there, you can produce large number of nascent oxygen molecule in your body, but because of this RECA activation, they will repair their DNA and the DNA replication will continue in spite of that oxidative stress. So RECA mediated gene repair is a very important pathogenic property of Streptococcus when the competence is on. Clear? Now we will discuss the different diseases caused by this particular Streptococcus pneumoniae. To start with, we have the sinusitis. Sinusitis, you know, the prenasal and paranasal cavity, they will produce the fluid and this fluid will accumulate. So the fluid accumulation in this particular sinus cavities will ensure too much pain, and that is introduced or induced by Streptococcus. Then we also have the typical infection of the middle ear. This is the middle ear where the infection is caused by Streptococcus and the disease is called otitis media. It is a dangerous disease because there is no major blood vessel there. So antibiotics will not treat this particular bacteria there. So it will lead to deafness sometimes, or damaging of the eardrum or tympanum membrane. This is the typical bacterial conjunctivitis. You know, the conjunctiva is the outermost shield of the eye. It can be invaded, and most importantly, there will be follicular conjunctivitis where there is lot of sebum production, and the condition may become even worse when there is viral conjunctivitis along with bacterial infection. It can also cause the Streptococcal Meningitis. That is, the meninges include the dura mater, pia mater and arachnoid mater. And these are the membrane of the brain which protect the brain. So if there is meningitis, there will be swelling of the brain. And because of the swelling, there will be internal pressure which will make the brain compressed. So it is a deadly situation we normally encounter in child. Meningitis is a common problem, or bacterial or streptococcal meningitis is a common problem in children. This is another important disease. You can see here the osteomyelitis or infection of the bone, which causes bone deformity, permanent deformity. The bone structure will be totally deformed. And if it occurs in the joints, this is a painful condition. This is the femur joint or the knee joint, which is causing septic arthritis, and the joint become totally dysfunctional. This is another important area where the bacteria can stay. This is inside the valves. You know, the auriculoventricular valves, that is the tricuspid and the bicuspid valves, they may show these are the red areas where the bacteria is growing. And this will grow and cause a disease called endocarditis. So just think, the bacteria can cause infection of the heart. It can also cause infection of the peritoneum. In the lymph node, it will spread infection in the small intestine, and the disease is called peritonitis. As I have said, it can cause cellulitis or streptococcal cellulitis in the leg, which is a reddening of the skin, but actually it is spreading along the nerve, spreading along the blood vessel, and the severe situation occurs in HIV patient, which can lead to amputation of the leg. But the most important disease is pneumonia, because pneumonia is actually associated with this streptococcus because the name is Streptococcus pneumoniae. And look at the symptoms. It can cause fever, it can cause skin infection. Of course, the most important area is the lung, where will be pneumonitis or pneumonitic patches. There will be muscular fatigue, there will be low blood pressure, there will be headache, there will be gastric irritation, all sorts of problem associated with pneumonia. And when this particular pneumonia is multi-drug resistant, then the situation is worst. Now, how can we detect pneumonia? Actually, there are specific symptoms with which WHO has actually helped us in detecting pneumonia. But primarily it is X-ray. As you can see, this white patch is the pneumonial patch in the lung which detects pneumonia. And as I was telling, the symptoms of pneumonia is very close to influenza. So a specific influenza kit is used to detect influenza bacteria, and if it is Haemophilus influenzae, then it will be positive, and if it is negative, then naturally this is the pneumonial infection. But as I was telling, that a definite infection of the lung can be in specific areas. It can be invading the lobe, so it is called lobar pneumonia. It can be invading the bronchus, it is called bronchopneumonia. It can be invading the interstitial spaces or different areas of the pleura, which is called interstitial pneumonia. Now, can you tell me that which particular person will be more prone to bronchopneumonia?
Sir, smokers generally predominantly suffer from bronchopneumonia.
Right?
Because they have an increased concentration of epidermal growth promoters.
Yeah, actually the nicotine present in the cigarette smoke or the hydrocarbon present in cigarette smoke, they will promote the epidermal growth regulation, or thereby they will be more prone to bronchopneumonial infection, and bronchopneumonial infection become worst in smoker. Apart from X-ray, you can also do CT scan of the chest or CT scan of the lung, and you can see this is the CT scan picture where you can see clearly the pneumonial patches. And this is another widespread pneumonitis. You can see these are the patches of pneumonitis denoted by CT scan. But how actually the person get the disease? It's a very common problem. We call it nosocomial infection. Usually hospital borne infection comes through the tube, that is the ventilated tube with which the person is breathing. So ventilation is actually the source of infection. Suppose you have admitted the patient for a different purpose, but he develops pneumonia. It normally comes through the ventilated tube. So it is called hospital-mediated pneumococcal infection or ventilator-mediated pneumococcal infection. So instead of saving life, it will make the situation worst. As I was telling, that there is a typical interaction between different bacteria invading the respiratory tract. It includes the Haemophilus influenzae, it includes the Neisseria meningitis, it also includes the Streptococcal pneumonia. There are specific tests, but when we see their development, you can see clearly Haemophilus influenzae, initially the growth is very high. Gradually it comes down, but you can see here this particular growth of the Streptococcus pneumoniae initially was high, then gradually it will appear as if it is not there, but it actually persists. And it persists for a prolonged period and cause the deadly situation at a later stage. Now, how to treat this particular or to prevent this particular infection? Definitely there has been a specific vaccine, and this vaccine, which is commonly called the pneumovax, is given only for pneumonitis cases, but unfortunately there are only say about 80 different strains are there, about only 20 to 23 strains are sensitive to this particular vaccine. So even if we take the vaccine for pneumonia, it is not guaranteed that we will be protected from pneumonia. Apart from that, so when there is a risk involved, we should prevent it. So a very ideal way of preventing is using typical air purifier system in our home, and thereby the air is pure what we take in or in our working place. We can also have typical washing of our hand after every meal or after any laboratory work. We should wash it properly with typical detergent, and that will remove the surface adhering pneumococcal bacteria. We can use surgical mask, because I was telling you this particular bacteria enters to the respiratory tract. So that is again very common. We should prevent it through surgical mask, particularly when we are going in busy roads, when there are a lot of people. You never know which person is carrying this particular pneumococci. And as I was telling, in HIV patient, the person is showing dreadful infection. So there may be different ways of treating HIV and making the person less prone to pneumococcal infection. And this HIV treatment, as you know, can be multi-drug therapy, which include the integrase inhibitor, which include the reverse transcriptase inhibitor, or which include the protease inhibitor. So as you can keep the HIV patient under control or HIV infection under control, pneumonitis in that person will be reduced. Of course, in a child, the best diet is the mother's milk, and mother's milk prevent the child from pneumonitis at the early age, or even, as I was telling, the infection of the meninges or meningitis or meningitis in the child. But if there is a particular antibiotic to be given, because we have already suffering from the disease. So what antibiotic we should give? We can see that there may be different antibiotic, of which cotrimexazole is an important one. Now cotrimexazole actually prevent the DNA synthesis, and it is a sulfur drug. So precisely those person who are sensitive to sulfur drug cannot be given cotrimexazole, but otherwise more or less cotrimexazole is seen to work in pneumonitis infection.
Sir, I have a question. Why cotrimexazole can be effective in pneumonitis treatment?
You see, cotrimexazole is not a single drug. It consists of sulfamethoxazole and trimethoprim. You can see here, sulfamethoxazole is blocking dihydropteroid synthetase, and trimethoprim is blocking dihydrofolate reductase. So when two enzymes are blocked, definitely this particularly tetrahydrofolic acid will be prevented, and thereby thiamine and purine synthesis will be blocked. So thereby we can say, instead of one drug, two drugs are there, and thereby it is effective. And more or less, because sulfur drug being a cheaper drug, it is less in use, so the bacteria is less exposed to this particular compound, so it has still effectivity in treating pneumonitis. Clear?
Yes, sir.
Now coming to infection in neonatals. In neonates, it has been seen the infection comes to mouth. So ideally, if artificially we can suck out, or mouth suction procedure is employed, then the bacterial flora can be taken out, and thereby the effect of pneumonitis can be reduced. But it is a risky phenomenon because the child is newborn and anything and everything can happen because of this suction phenomenon. But this is a very good drug which is commonly called amoxicillin clavulinate combination. This amoxicillin clavulinate is cell wall biosynthesis and also penicillinase inhibitor. So naturally the combination of these two drugs, which is commonly called say augmentin, which is a very common drug used in pneumonitis. But these days we have seen that even this particular streptococcal pneumonia through hospital, they have actually developed resistance to augmentin or amoxicillin clavulinate combination. In those cases, we have only one option, which is use of meropenem or imipenem. So perhaps this is the most costly and the latest molecule which is being used to treat the Streptococcus pneumoniae infection. So in this particular lecture, what we have tried is, we have tried to discuss the different aspects of Streptococcus pneumoniae. We have introduced its discovery. We have introduced its cell structure, its genome pattern. Of course, I have also discussed about their transformation. I have discussed that in which particular areas they are distributed. And then finally, I have discussed the different ways by which this particular dreadful infection can be controlled. Okay? Thank you.